Good day, everybody. I'm Jackie van der Wals from the Department of Plant and Soil Sciences at the University of Pretoria. This webinar deals with late blight on potatoes. I'll touch on the symptoms, epidemiology, and management thereof. So the contents, as I've mentioned, will start with the symptoms. I will discuss the pathogen with you, go through the disease cycle in some detail, and then briefly touch on some management options for this disease. Let's start by looking at the symptoms of late blight on potatoes. So let's start our way from top left and work in a clockwise direction. The first symptoms of late blight on potatoes are noticed as small light to dark green, circular to irregular shaped water-soaked spots, and these normally occur on the lower lesions on the denser parts of the plant canopy. On the lower leaves. When conditions are favorable for the pathogen to develop further and cause further disease, and these are cool, moist conditions, these lesions will expand and sometimes they appear greasy, as you can see in the second photo on the left. And in many cases, they will have a chlorotic halo around the lesions. One often also sees black lesions on stems, as you can see in the photo on the top right-hand side. A characteristic sign of Phytophthora infestans infection on potato is a white mildew-like sporulation under favorable conditions. Here you can see this white mildew-like sporulation on leaves and on stems of a potato plant. So as I've said, this is quite a characteristic property and it helps to distinguish Phytophthora from other pathogens. Under severe conditions, lesions will also occur on tubers. The tubers could either have relatively hard rot with indentations on them, or they could have a granular brownish colored rot, which is seen under the skin of the tuber. Late blight is often easily confused with botrytis grey mold on potatoes. So I've given you some photos of botrytis grey mold. The primary difference here is that botrytis often forms concentric rings on the leaves, which late blight does not do. But the other most prominent difference is that the sporulation of botrytis is a dark brown to a grey sporulation, whereas that of Phytophthora is white. Another kind of injury which could also be confused with Phytophthora is that of a frost injury, as we can see in the bottom right-hand photo. But if one knows the conditions that surrounded this injury, one can easily rule out late blight, and it would be easy to see that this is frost rather than a disease. Let's have a look at the pathogen. It's quite an infamous pathogen. Phytophthora infestans, it's something that all first years and second years learn about when they study plant pathology. In fact, if we break up the name phyto and thora, it actually means plant destroyer. So this is literally what this pathogen is. It destroys plants and it does that in a very short period of time. So Phytophthora infestans is not a true fungus. It's not actually grouped with a fungi. It's what we call an oomycete or a water mold. It's a very aggressive pathogen and it is also able to infect plants directly. It does not require a wound or a natural opening for infection of plants. What's important to know about Phytophthora is that there are two main mating types, the A1 and the A2. And at present, only the A1 has been detected in South Africa. We do need to be aware, however, that the A2 is steadily moving down south in Africa. The last survey that was done a couple of years ago found the A2 in Kenya. However, as I've mentioned, in South Africa, we currently only have the A1, and it is very wise to keep it this way. These A1 and A2 mating types are not to be confused with the different strains of Phytophthora. So if you read the literature, you will find that there are many different strains, and these refer to where they were isolated from, what type of resistance or what types of cultivar resistance they have overcome, etc. What we need to know about A1 and A2 is that where only one mating type is present, as we currently have in South Africa, only asexual reproduction occurs. And this is by means of sporangia, which are produced on stalk-like sporangiophores, as you can see in the picture there. And these sporangia are dispersed by air or by wind-blown rain to neighboring fields and neighboring plants. Where both mating types are present, however, sexual reproduction occurs, and this results in the formation of sexual oospores by meiosis, and these are thick-walled and can survive in soil. So in South Africa, because we do not have both mating types, we do not have survival of oospores in soil. Let's move on to looking at the disease cycle. As with early blight, late blight is a polycyclic disease. This means, firstly, that secondary inoculum is produced through the season, and this inoculum allows the pathogen to complete more than one disease cycle, many disease cycles, in a growing season of the plant. So you can see in this disease cycle that secondary inoculum is produced here. If we have a look at the source of primary inoculum of Phytophthora infestans, and this is in the absence of oospores, in other words, where there's no sexual reproduction occurring, mycelium and sporangia that are found either on infected tubers, which are left lying in the field after harvest, are a source of inoculum. The other sources of inoculum are sporangia, which are produced on volunteer potatoes or on other host plants found in the area, such as weeds. Infected seed tubers that are planted also constitute a source of primary inoculum. So we're now going to look at the cycle of where secondary inoculum is produced, which is essentially the cycle that causes the main epidemics. And for the purpose of this talk, we are going to ignore the cycle where oospores are formed, because in South Africa we do not have this cycle occurring. It's very seldom that, or it occurs very rarely in nature in any case. So we're focusing on this part of the disease cycle, the sporangial formation and germination. So sporangia form on lower leaves and stems, generally in a dense canopy, where the relative humidity is high but not much higher than 90%, or where free moisture has occurred for 12 hours or longer. Sporangia can form at temperatures between 3 and 26 degrees C, which is quite a wide range, although the optimum temperature for sporangial formation is between 18 and 22 degrees. Now, once sporangia have formed, they can penetrate or infect the plant in one of two ways. Firstly, they could either germinate directly, as you see here, through a germ tube, and this occurs at temperatures of 21 to 26 degrees, so that's not particularly cool. Or sporangia can develop zoospores inside them, anything from 6 to 8 zoospores in one sporangium, and this occurs at temperatures lower than 18 degrees C. So if you have lower temperatures with moisture, one will find the production of zoospores. When the temperatures go up, then you know that the sporangia are going to germinate directly and infect plants via the germ tube. Now this is important because if we consider that when zoospores are produced, each zoospore is a probigule for infection. So in other words, the number of propagules for infection is multiplied between 6 and 8 times, because each sporangium produces 6 to 8 zoospores. So this is why epidemics of late blight normally occur at the cooler temperatures, because zoospores are produced, and these zoospores in free moisture can then swim to infect other hosts. So here we see the sporangium producing zoospores, releasing the zoospores, and the zoospores then infecting the plant. The sporangia are carried or dispersed by wind or by rain-splashed wind-splashed rain to healthy plants. They can also be washed into the soil to infect the tubers in the soil. We see this down the bottom of the disease cycle. Now this disease cycle, in other words, from development of a sporangia for production of sporangium and reinfection of the plants, occurs once every 5 to 7 days under favorable environmental conditions, just demonstrating how rapidly this disease can develop. After infection, symptoms are noticed within a few days, probably as little as 2 to 3 days. One can see infection one can see symptoms after infection. So we're taking it back to the total disease cycle. If we can eliminate primary inoculum from the beginning of the disease cycle, we can fairly well control late blight. If, however, the primary inoculum is not eliminated, then one needs to go to looking at ways of eliminating the production of sporangia and zoospores. So this is where management comes in. As with any other pathogen, the best management is through an integrated approach. So putting together a whole lot of different approaches. It's not just one silver bullet, not just applying a fungicide that is going to manage late blight. So firstly, to destroy or to remove primary inoculum, one needs to destroy cull potatoes, remove all volunteer potatoes, and ensure good weed control. It's also wise, as I always recommend, to have a 3 to 5 year rotation with non-hosts, so that there's no host debris harboring the pathogen in the field. Of course, critically important to plant disease-free seed tubers. That said, it is not impossible that the pathogen can still be found latently in the present or in disease-free seed tubers. So even if they are certified disease tubers, there is a certain tolerance, and it's possible that the pathogen could be disseminated via seed tubers. However, the chance is much lower if one plants certified seed tubers as opposed to uncertified seed tubers. Site selection or field selection is important. One should always plant in well-drained fields, because the more water that is lying in the field, or the more waterlogged a field is, the greater the chance of tuber infection and spread of the pathogen through the soil moisture. Irrigation scheduling is important, as it is with early blight, to ensure that the plants do not stay wet for too long, and to ensure that the soil moisture drains to such an extent that we don't have waterlogging. Plant nutrition again plays an important role. As always, it's important not to over-fertilize or to under-fertilize. So one needs to know exactly what the plant needs and not to put too much down or too little. In South Africa, the trend is, well, probably worldwide, the trend is normally to give too much fertilizer, and that is not a good thing. To suppress the development of late blight, one should reduce the amount of nitrogen and increase the amount of manganese and potassium, but again, not too much. Cultivar choice, although important, we need to bear in mind that not all cultivars have a level of resistance or tolerance to Phytophthora, and it's not always possible to choose cultivars that have some resistance to this pathogen. However, if Phytophthora is a problem in the growing region or in the area that you're planting in, this would be a starting point to choose cultivars with a level of resistance to the pathogen. Scouting and roguing is important. One can scout for the disease, and when one sees contaminated or infected plants, remove them from the field, dig up the tubers, and take out that source of inoculum. And then, this is the most common method, but as I've said, one can't just rely on a fungicide spray program. But obviously, Phytophthora is fairly well controlled in South Africa with fungicides. One can also use biocontrol agents to suppress the disease, and these also play a major role in strengthening the plant and increasing the plant's immunity. As with early blight, it is important to alternate active ingredients from different frac groups when applying a fungicide spray program to prevent resistance development in the pathogen.